What an honor to be here today and discuss our scientific accomplishments in the laboratory. I'm inspired by what I've heard so far, and maybe just briefly we'll share a little bit of my personal story. Next year, actually I hadn't thought about this until this morning, but next year will be the 50th anniversary of my coming to Los Angeles and to California. We came here as immigrants from Beirut, Lebanon in 1976. My parents would always say, like many of you, 5 people, one suitcase, no money. But California gave us opportunity to have an opportunity. And a few things happened, and now I'm a professor at Caltech. And I'll tell you about that work that we've done in the meantime. The work will be a little bit technical. I'll try and walk you through some of the jargon and some of the scientific intricacies of what I do, but hopefully you'll understand both the specific details of this research program, but also more broadly what we're trying to accomplish in really trying to understand how the body works and how we can intervene in situations where there is disease. This is the Turtle Pond at Caltech, about 20 miles from here. And the title of the talk is essentially about Parkinson's disease and what we've done as microbiologists to try and understand this classical neurological condition, and again, develop novel therapies to address the unmet need of Parkinson's disease. Just to orient us, the tissues that we're excited about in the laboratory are the gut and the brain, the intestines, the stomach, small intestine, colon, and how it communicates with the brain. And this is bidirectional. The communication goes in both directions. I won't walk you through the details, but suffice to say, there are neurons, nerve cells that connect the two tissues, small molecules, hormones, immune cells that are trafficking between the gut and the brain. So these two organs are in constant communication. It's really this super highway between the intestines and the brain that we hijack, if you will, to try and understand how we can essentially get improvements in brain health by drugging the gut, by improving the function of the intestines. So our laboratory over the years has worked on a number of different disease models. We've worked on inflammatory bowel disease. You may know as Crohn's disease, ulcerative colitis. We've worked on multiple sclerosis. More recently, we work on autism and depression. But Parkinson's caught our eye about 15 years ago because of the really interesting and even perplexing correlation between gastrointestinal disease and brain disease. The vast majority of individuals with Parkinson's will have constipation, clinical grade constipation at some point in their lives, most certainly, about 80% of them, most certainly before they develop the motor symptoms. And so you can see on the lower graph that by the time a person is diagnosed with Parkinson's, which the hallmarks of which are essentially movement disorders, tremors, posture instability, and gait issues, that the vast majority of those individuals would have liked that constipation for years, if not decades, before they were diagnosed. There's other prodromal or preceding symptoms that, again, this was quite confusing to me as a microbiologist, but someone who was studying the gut at the time, and really known in the neurological community, but entirely understudied of why, again, this neurodegenerative brain disease has gastrointestinal manifestations. So like most things in biology, the history goes back decades, in this case centuries, to this essay written by James Parkinson in 1817. At the time it was called Shaking Palsy. It was named after him by others. And in reading this article, it caught my eye that he was essentially going through case studies and talking about people with tremors, and noted that the majority of those individuals had essentially what is constipation, what we call constipation today. And in at least a handful of those individuals that he was following, he gave them a bowel cleanse and reported that the bowel cleanse improved their motor symptoms. And so again, the seeds were planted many centuries ago. And more recently, there's been about 20 years ago, a hypothesis, it's called Brock's hypothesis, that indeed a certain percentage, maybe the majority of Parkinson's disease may originate in the gut, meaning that the actual disease starts in the gut and then travels to the brain over time. And maybe that's what explains why the GI symptoms, the constipation, precede the motor symptoms. This is a hypothesis. We, others around the world, are testing this. And I think by the end of the talk, you'll see why we think there's value in this. But
Suffice to say, again, there's several, dozen publications, some from humans, mostly in animal models, that suggest that indeed one can experimentally initiate Parkinson's disease in the gut and then get the classical motor symptoms of PD. And Parkinson's is now the fastest growing neurological condition in the world. And so this is a major social, economic, and medical problem now, and it will continue to be so in the decades ahead. And so, again, as microbiologists, we wonder what's happening in the intestine in terms of the microbes that inhabit the lower gastrointestinal tract, and whether or not these organisms, these small microbial cells, are indeed part and parcel of the pathogenesis of Parkinson's disease. And so just very quickly, some of you may know this, but I still think it's quite fascinating that we as humans are mostly bacteria. We are outnumbered by bacterial cells about 3 or 4 times. And so our only cells are actually dwarfed by the number of microbial cells. Our immune systems are tuned by these organisms. Our metabolic system, our nervous system are essentially functioning the way they do because of the signals these organisms send into our bodies. The brain and liver are approximately the same in terms of both function and size of the microbiome. The metabolic capacity of the microbiome equals that of the liver. The size of the microbiome equals that of the brain. Mostly in animal models that include the majority of which are mice, but other rodents and non-human primates and a few other species, there's almost no dispute at this point that the microbiome is contributing, in some cases, causing neurological, metabolic, and immunological conditions. And a lot of that is shown here. What we still don't understand in humans, we have some correlative evidence, we still don't understand in humans, are these same principles driving disease in people? And again, that's one of the central questions that we and others in the field are asking. And so a few years ago, we asked a very simple question: is the microbiome different between individuals with Parkinson's and healthy controls? And so our tissue of choice is stool. We heard a little bit about sanitation earlier, and so there's a bit of a link to that. But ultimately, it's an accessible tissue, and I think there's a lot of information in stool, as much as there's an ick factor as well. But we essentially collected stool from individuals, a couple of different cohorts, one that was based in Chicago, the other that was sampled from around the country, and using essentially advanced DNA sequencing, we mapped all the microorganisms that are in the Parkinson's microbiome, the microbiomes of individuals that live in the same household, essentially spouses, and the population in general. And so again, this is data that may be hard to deconvolute, but if you just look at the gray bar or the gray symbols, that's where the aggregates sequence or profile of the microbiome lands, and in PD, those gray bars are different than that of healthy controls, meaning that the composition of the organisms, both their abundance as well as their representation, is altered in the stool of individuals with Parkinson's disease relative to that of healthy controls, again, whether they're a household or a population. And of course, this is getting into the weeds a little bit, but there are very specific organisms that are stereotypically different in Parkinson's versus those in the population. I think this gives us clues as to what organisms may be either driving disease or maybe protective from disease. And again, this is actionable for us. We're now following up on some of these organisms that we identified and testing them in the animal models to see if we can actually understand what these microbes are doing in the context of whether it's affecting the function of neurons, affecting the function of the immune system, the metabolic system, the endocrine system, and so on. So it's widely known that Parkinson's disease is caused by aggregation of a protein called alpha-synuclein. We all have this protein. It's an evolutionary conserved protein. It goes down to flies and worms, and it's involved in neuronal function. It's part of our daily lives. When you make a decision, when you exercise, alpha-synuclein is doing its job in neurons. Unfortunately, alpha-synuclein has a very unusual property, is that it aggregates, it clumps, it forms these hairballs inside of cells, if you will. And so that is like sand in a gear. It will essentially affect the function of cells. Cells become dysfunctional. They work very, very hard to perform their function. And if these aggregates, these clumps of alpha-synuclein are not cleared, eventually those cells die. and that's neurodegeneration. So again, as you likely know, Parkinson's disease, like Alzheimer's disease, is a disease of neurodegeneration. Neurons in the brain, neurons all around the body die, and they die because this protein is not cleared in time and effectively stresses the cells until they ultimately are lost. And so we work, as I mentioned, with animal models, specifically with mice, and you can model many, many features of Parkinson's in rodents. This animal model overexpresses human alpha-synuclein. It has many of the same symptoms, many of the pathophysiology of Parkinson's disease. It gets motor symptoms. It has GI symptoms. And before we started doing this work, no one had ever asked the question, what's the role of the microbiome? So a very simple experiment that we did that we ultimately published in 2016 was, if you remove the microbiome of mice, how do they perform in standard Parkinson's disease testing? So the first test I'm showing you is a challenge beam. We just take a mouse, put it on a platform, and measure the time it takes to cross the beam. The first two bars are mice with essentially a lab microbiome, several hundred organisms, mouse species that are living in their intestines. And you can see that the alpha-synuclein overexpressing mice, ASO, that second bar, those mice take longer to cross the beam than their wild-type counterparts. These mice have motor deficits, again, modeling what you see in human Parkinson's. In the next two bars, these are the same exact mice, but are missing a microbiome. We just essentially depleted all their microorganisms. Everything else is identical in these mice, and you can see that these mice no longer develop motor symptoms. It's true for a second test, a pulled scent test, and again, I'll go quickly because you get the point, in an adhesive removal test. The first two are tests of coarse motor skills. The final one is a test of fine motor skills, where we essentially take an adhesive, put on the nasal bridge of the animal, and score the time it takes to release or to grasp and release that adhesive. And you can see there's a defect in mice that are colonized with microbiome, but removing the microbiome alleviates those symptoms. It's the same with GI symptoms. We're essentially looking at fecal output, number of fecal pellets that the animal produces in a given amount of time. And you can see in the upper right-hand corner that these animals, if they have a complex microbiome and are overexpressing synuclein, produce fewer fecal pellets than mice that are missing a microbiome. And in the bottom panel, you can see that the pellets themselves are drier. So essentially, we're modeling constipation, fewer bowel movements, the material itself is drier. And again, these features are regulated by the microbiome. Alpha-synuclein aggregates in the brain are, again, what cause those motor symptoms. So we wanted to understand, if we remove the microbiome, what is happening to this protein? What's happening to its pathology? And so you can see in the SPF mice, again, these are mice with a complex microbiome, that there is widespread alpha-synuclein aggregates. That's the staining in red. You're looking at a tissue section of a brain region that's affected in this animal model. And again, this is the hallmark pathology. This is what's driving that neuronal stress that ultimately leads to neuronal death. In the panel to the right, this is the same exact animal without a microbiome and no alpha-synuclein aggregates. So something in the intestine is inducing aggregation of synuclein in the brain. And there's no evidence that the actual genetics of the animals is changed. The protein is made. It's made at the RNA level. It's made at the protein level. It just doesn't aggregate. And so again, this gives us fidelity in these experiments to understand that indeed it's the aggregation component, not just the production of the molecule, that's affected by the microbiome. So we did many, many additional studies to convince ourselves that indeed the microbiome was impacting motor symptoms and pathology in these animal models. Our results have been reproduced by a number of labs around the world. At the same time as we were doing these experiments, we realized that we have potentially a vehicle in mice to test human microbiome. So the data I showed you a few slides ago, where we're essentially profiling the microbiome of individuals with Parkinson's, gives us a snapshot into their organisms and the abundance of those organisms, and in some cases, the localization of those organisms. But it doesn't tell us that those organisms are actually doing anything, that those microbes are actually either protective or pathogenic. And so we found an opportunity that once we've cleared the mouse microbiome from this animal model, that potentially we can now test the function of human microbiomes. And so working with collaborators at Rush University, we were able to collect fecal samples from individuals with Parkinson's and household controls, and we transplanted microbiota intact fecal transplants from humans into mice and then tested the motor symptoms. These are the same 3 tests that I showed you earlier, the beam traversal, pole descent, and to the right, the adhesive removal. But in this case, again, we're taking both the wild-type animals and alpha-synuclein overexpressing mice and transplanting in either healthy control, that's the HC, or PD microbiomes. And you can see, just compare the second and the fourth bars, you can see that if the genetics of the animal is predisposed to developing Parkinson's, that the microbiome from a Parkinson's patient makes the symptoms worse than the microbiome from a healthy control. So again, this suggests that there's something functional in that microbiome, something about the PD microbiome that's dysregulated, that's exacerbating disease. And again, if we can find what those organisms and what those molecules are, we can drug those pathways. And so through many additional experiments, we identified, at least in a subset of individuals with Parkinson's, somewhere close to about 20% of individuals with PD, there is an overrepresentation of E. coli species that make a protein called curli. That curli fibers in the bottom right-hand corner, you can see this spaghetti-like structures around this E. coli cell. There's an actual image of E. coli are these curli fibers. So what's interesting about these fibers is that they're made of a bacterial amyloid. They're made of a bacterial protein which has many properties similar to that of alpha-synuclein. It aggregates, it forms these clumps. In fact, that's how these structures are formed, is that this bacterial protein is folding in a way, again, aggregating in a way to perform its normal function. And so we asked the question, can this bacterial protein induce alpha-synuclein aggregation in the gut? Is this the trigger in the intestine that's leading to the physiology, to the initiation, at least in this animal model, of Parkinson's in the intestine that then, again, moves its way up into the brain and causes the motor symptoms? And we looked in our data set, and indeed, the protein that encodes for these curli fibers is called CSGA. This is the bacterial amyloid, and it's elevated in the fecal samples from the PD patients and not from the healthy controls. And so again, this is partly data, but partly hypothesis that we tested. And over the years, a number of studies from cohorts all around the world have actually shown that our prediction, our guess, may likely be correct, because once they went back and looked for this particular protein in different data sets, this is a study from Korea that was just published last year, that there's elevation of these bacterial genes, elevation of the curli encoding genes in individuals with PD relative to that of healthy controls. Again, a nice correlation, more refined than the previous studies, but suggesting that indeed just increasing levels of this bacterial protein may be a risk factor for Parkinson's. And so then we went back to the animal model and we asked, if we now put in one organism, an organism that makes this bacterial amyloid, this wild-type E. coli strain, can we induce motor symptoms? So I draw your attention to the data in the white bars. Again, very similar to the data I showed you when the mouse had a complex microbiota, but again, in the presence of one bacterial species in these mice, this one single organism is able to induce all the motor features of Parkinson's. And the blue bars is the exact same organism that's deleted in CSG, deleted in the curli fibers. And so the organism is making all the other proteins, all the other molecules that E. coli makes, but is missing just that bacterial amyloid, and you see that these mice no longer develop motor symptoms. Same with the pathology. So when we look in the brains of these animals, again, this is the most critical analysis in my estimation is there synuclein aggregation in the brains of these mice? The aggregation levels are entirely controlled by the presence of this bacterial protein in the gut. The more of bacterial protein, the more aggregation of alpha-synuclein in the brain, both in regions of the brain that are affected for movement, but also regions in the brain that are affected for reward and other physiological contexts. We showed for the first time in this study that indeed this bacterial amyloid can also induce aggregation in the gut. Again, going back to Brock's hypothesis, that potentially the seeding of synuclein begins in the intestines. Can it be done by this bacterial protein? And we show that the CSGA protein, again, can induce synuclein aggregation in the gut. So our working hypothesis is that in this animal model, the way we set it up, that the disease begins in the intestine and spreads through the nervous system of the intestine, called the enteric nervous system, and then gets on essentially the superhighway, the neurons connecting the intestine to the brain, called the vagus nerve. And then once this pathology is in the brain, then it spreads throughout the brain and over years makes the symptoms worse in Parkinson's. And so based on that work, we then became a little bit more ambitious, if you will, and said, if this hypothesis is true, can we intervene at the stage of the gut? Instead of trying to get drugs into the brain, can we develop drugs that actually target this bacterial amyloid, begin early or intervene early in the process of Parkinson's and prevent or even treat the symptoms of PD? And so I started a biotech company in 2016 to essentially take this academic work into the clinic. And one of the first activities of the company was to develop a small molecule drug that prevents the bacterial amyloid from folding. Because if the bacterial amyloid itself can't aggregate, then it doesn't induce alpha-synuclein to aggregate. And so we developed a drug called AX5006. Again, it's an oral small molecule. What's interesting about this molecule is it doesn't cross the gut barrier. It stays entirely in the intestine. It doesn't even get into the blood, let alone the brain. So this is revolutionary in many ways because every drug currently being developed for Parkinson's or Alzheimer's or depression or sleep issues, their barrier is to get drugs into the brain. We're just using biology to signal from the gut to the brain. These drugs only enter into the gut. They're not absorbed. And so not only is there proof of concept using this type of drug, but there's also less side effects. You just don't see some of the off-target effects that you do when drugs are getting into the brain. And so we showed just in vitro, this is just purified proteins, that alpha-synuclein aggregation is reduced by AX5006 in a dose-dependent manner. And this is the data I was telling you about, that when we orally treat animals, in this case it's mice, but we've now done this with dogs and pigs, that oral administration of this drug results in the drug staying entirely in the gut as measured in the feces, but not in the serum and not in the brains of these mice. And so not only does AX5006 reduce alpha-synuclein, it actually does what it was intended to do, and that's prevent the aggregation of CSGA, that bacterial amyloid. And you can again see this is a biochemical assay, just purified proteins in the test tube, and the aggregation is inhibited. And what's also interesting is that when we now put this drug into mice that are overexpressing CSGA that make this bacterial amyloid, the levels of the bacterial amyloid are reduced. So it has all the features that we would want from a drug that's hitting its target. The drug has no safety issues, has no toxicity to the best of our knowledge. In fact, this drug will be in the clinic later this year. But most importantly, we want to understand, can we alleviate motor symptoms? Again, this is what patients suffer from, and this is our goal. And so again, using the same behavioral tests that I've already introduced, you can see that almost in a dose-dependent manner, treating these animals with this oral drug, with this small molecule, not only prevents the formation of motor symptoms, which I'm not showing you here, but actually reverses the motor symptoms. So these animals are aged to a point where they manifest disease, and then we start orally dosing the animals with the drug. And you can see in the beam traversal, the pole descent, and in the adhesive removal test that mice that already had disease no longer show symptoms being given this drug orally. And again, this is only about 10 weeks of treatment. And so we're quite excited about the potential of actually getting this type of improvement in the clinic. And then importantly, that hallmark pathology of alpha-synuclein aggregates in the brain are also resolved by this drug. Again, we're drugging the gut, not the brain. But you can see that these preformed aggregates, this pathology, this plaque that's forming in the brains of these mice, are resolved, are entirely cleared by giving the animals this oral drug. And so I'll leave you with a couple of concepts here, is that whether it's the genetics of Parkinson's or the lifestyle, the diet, factors that we don't really understand, the microbiome of PD patients is different than that of their matched controls. And it's really this difference, this change in the microbiome, this reconfiguration of organisms and what they're doing in the gut, then signals to the brain to cause disease, and that we can intervene at the level of the gut with drugs. We can intervene at the level of the gut with diet. We've already published several studies showing that diet, depending particular types of diet, are therapeutic in this animal model. And then we're now working on other modalities such as fecal transplants to see if essentially a healthy fecal transplant into mice will also give us these same improvements in symptoms. And then the concept that essentially drives a lot of our thinking, both in the lab but also commercially, is this notion that maybe drugging the gut, delivering these therapeutics to the intestine is much easier than that of getting drugs into the brain. And again, if this gut-brain connection is mediating health outcomes or negative health outcomes, then we can essentially just deliver drugs to the microbial targets and get long-term benefits without, again, the challenges of having drugs cross the blood-brain barrier and into the brain itself. And I'll leave you just with the most important slide. The people who did the work, Matthias, Livia, and Tim are all former or current postdocs, PhD fellows, and technicians who really spearheaded this work. They did all the work that I present to you today. In green are the collaborators, in yellow are our collaborators, and in green are funding sources, and Axial is my disclosure. Thank you.